Gene expression begins when a signal binds a receptor on the outside of the cell. In this case, the cell is a cell in the apial ectodermal ridge of the AER, which is on a developing limb. A signal is sent to the inside of the cell, where it goes through the transduction cascade, which is a series of 3 enzymes that modify the signal until it is ready to bond with the signal receptor on the outside of the nucleus. The signal on the outside of the nucleus sends a transcription factor to the promoter of the gene, which is the FGF4 gene. The transcription factor, in combination with the promoter, attracts RNA polymerase. And the RNA polymerase unzips the gene. So here it is on the promoter, and it unzips the gene. It unzips the gene and starts building a transcript pre-mRNA off the template strand of the DNA. A cap and a tail are added, and the a cap is added, which is a modified form of guanine, and a tail, which is a polyadenine, is added. The RNA polymerase keeps doing its thing downstream until the 5' stop signal. Then some SNRPs form a spliceosome, and the spliceosome spliceosome removes all of the introns, which are going to stay in the nucleus, and pastes all the exons together. So you end up with this. It's now ready to leave the nucleus. It's called mRNA. So it leaves the nucleus, and it's hanging out in the cell. And a tRNA carrying the amino acid methionine shows up and bonds itself to the start codon of the mRNA. Then it is attracted to it because of its specific anticodon. Then a small ribosome unit slides into place, if Lily can find her finger, and a large ribosome unit attach themselves. Then a whole bunch of other tRNAs all kind of go through, and they all are carrying, and all the tRNAs that go through eventually will carry all the amino acids. All the amino acids will pass through the strand. They each carry a different amino acid. They make the FGF4 protein, and that protein is released by the cell, and it goes all the way off in its lonesome onto Lily's rug, or slash the rest of the body, out of the cell. The FGF4 cell is sent to a cell in the ZPA, where it bonds to the ZPA cell, and it triggers the expression of the Sonic the Hedgehog gene, which was responsible for the creation of the posterior structures in a vertebrate limb. So when the Sonic Hedgehog gene is triggered, it begins production of a posterior structure in a vertebrate limb, starting to code for the arm.